Okay, so I'm pretty tired, which is probably not a good way to start the video, but I've got my coffee and I've got my tea. Yes, that's how tired I am. So I've got both going on here. Anyways, for the topic of discussion today is gene regulation. So what regulates in prokaryotes and eukaryotes? What's going to regulate what genes are being expressed? And you're going to hear this term a lot, gene expression. What does that even mean, gene expression? It just means that the gene on a chromosome is being transcribed and translated, which is something that we just recently talked about. So if a gene is expressed, then the protein is being made and the cell is making that protein. That's what we mean by gene expression. And so in this chapter, we're going to be addressing this big question, if all of our cells contain the same genetic information, how do cells differentiate, meaning how do they become different, and how do they perform completely different specialized functions? So we have all these different kinds of cells in our body, like muscle cells, fat cells, bone cells, blood cells, nervous cells, epithelial cells, immune cells. They all have the same genome, they all have the same genetic information, but what makes them so different? How did they differentiate? How did they become so different? And it has to do with gene expression, okay? Gene expression. So what makes those cells different is that they are expressing different genes. So in an epithelial cell, so on the right-hand side over here, you can see that on an epithelial cell, the genes that code for epithelial proteins, those are the genes that are being expressed. Whereas with the neuron, the genes that are specific to a neuron, so the neuron-specific genes, those genes are being expressed. In a neuron, they're making neuron protein, okay? All right, and in prokaryotes are very simple organisms. Many of them have a very short lifespan, maybe 20 minutes. So they're single cells, and because they have such a short lifespan, they have to quickly respond to their environment. Like let's say that if food sources is available, then they need to start making the enzyme that's going to break down that food source very quickly. They need to be able to exploit transient resources. So if we're going to be talking about today, like if there's lactose available, it needs to make enzymes that are going to break down the lactose. If there's glucose available, then it's going to shut down the gene for enzymes that are going to break down the lactose. Sorry, I'm getting distracted because there are like these two squirrels outside just like chasing each other around. Okay, anyways, and then so gene regulation is like turning on or off these genes very rapidly, and prokaryotes or bacteria are very good at that. There is a very simple on and off switch, which we'll be talking about in the Operon model, that allows for them to turn on genes very quickly, turn off genes very quickly, okay? And so there are two ways that bacteria can control the way that a metabolic pathway is controlled by enzymes. Okay, so bacteria can easily control gene expression by regulating transcription in two ways. Well, actually, the first option is not a way that they're going to prevent transcription from happening, but we do call it a way of controlling gene expression. Because it is controlling a metabolic pathway. So here we have a metabolic pathway. We have a molecule that is a precursor. So let's say that is molecule A. And molecule A is a reactant which is going to turn into the product molecule B with the help of the enzyme 1. Reactant B is going to become product C with the help of enzyme 2. And then reactant C is going to become the final product, which is tryptophan, with the help of enzyme 3. So this is a metabolic pathway which utilizes 3 enzymes to go from precursor A to the final product, which is tryptophan. And tryptophan can act as an allosteric inhibitor. It can go back, and it's called negative feedback, right? By negative feedback, it can go back up to this first step and inhibit enzyme 1. It's going to act as an inhibitor. I hope that you guys remember how those inhibitors work. Remember, allosteric inhibitors are going to bind elsewhere other than the active site. They're going to bind somewhere else on the enzyme, change the active site, change the shape of the active site so that the substrate, in this case the precursor, can no longer bind. Okay, so if if this is enzyme 1, and I like to always draw it simply shaped. Okay, so if this is enzyme number 1, so E1, and there's tryptophan, tryptophan is going to bind somewhere else other than this active site. That's the active site, and it's going to cause the active site to change shape. And when it changes shape, precursor or substrate A can no longer bind to the active site, and then we're not going to get substrate B, and then substrate B will not be able to turn into C. So this is called negative feedback. If the bacteria is producing tryptophan, which is an amino acid, I forgot to mention that. It's an amino acid that's very important to make proteins, okay? And so if there's enough of that tryptophan around, it's making enough tryptophan, then it's going to shut down this whole metabolic pathway to produce tryptophan. And that makes sense, right? Because if you have enough of something, why make more of it? So this is one way for the bacteria to conserve energy, I guess. It's conserving energy because it's not wasting enzymes. It's not wasting energy to produce something that it doesn't need. The second way that, so number 1, product of metabolic pathway acts as an inhibitor. Okay? The second way that prokaryotes or bacteria can regulate gene expression, control gene expression, is the tryptophan is going to actually act as like a repressor or shut down these genes. So these genes are not going to go through transcription. They will not go through translation, and you're not going to end up with an enzyme at all. Okay? So this pathway is where tryptophan itself is going to go to the DNA. It's going to go to the DNA where those genes are found, and it's going to shut down those genes, switch off those genes so that you do not get enzyme 1, enzyme 2, or enzyme 3. So I was telling my classes today that, this way, okay, the second way is energy conserving. It saves the most energy because the cells, the prokaryotes, the bacteria, they're not spending energy making the enzymes. Whereas in the first way, they do make the enzymes. It's just that tryptophan shuts down the ability for enzyme 1 to do its job. Therefore, you're not going to get it shuts down the entire metabolic pathway by inhibiting enzyme 1. But the enzymes are still being made in this way. Okay, so really shutting down the genes is the most efficient, the most energy conserving way that bacteria can control gene expression. So how do bacteria shut down these genes? Okay, so let's talk about that. They have operons, okay? So operons are only found in prokaryotes. So that's one misconception that I see a lot with students, is that they think that eukaryotic cells, such as our cells, we have operons. We do not have operons. We do have promoters and we do have genes on our chromosomes, but we don't have an operator. We do not have repressors or regulatory genes, okay? So the Operon model, this is what we call the Operon model. It is only found in prokaryotes or bacteria. So what is an operon? An operon has a promoter. Remember, the promoter is where RNA polymerase will bind to. That's going to unzip the DNA and transcribe these genes. Then you have the operator. The operator we would consider as the switch. Okay, the switch. This is the on or off switch. And then here are the structural genes. When they get transcribed, then they get translated, and then the amino acid or polypeptide chain or the protein is made by the bacteria. And then just outside the promoter, you have the regulatory gene. The regulatory gene is going to code for a repressor. Sorry, it's going to code for the repressor. Okay, and the repressor protein that it codes for does exactly what it sounds like. It's going to switch off these genes. Okay, the repressor is the protein that's going to switch off the genes, repress it, okay, stop it from being transcribed. So there are two operons that you absolutely need to know. Okay, you definitely need to know the two operons. The trip operon in bacteria is known as a repressible operon. Okay, so there's a regulatory gene, as I said, and it codes for a protein called the repressor. So here we have an mRNA, so it's been transcribed, and then it gets translated. Okay, so this is transcription, this is translation. It doesn't make sense to abbreviate transcription this way, right? I need to actually write it out. So we have transcription, translation, and then you get the repressor. Okay, and here's our RNA polymerase. It binds to the promoter, and it goes past the operator. Remember, the operon is the you have the promoter, you have the operator, and then you have the genes that are going to be expressed. Okay, so there's our RNA polymerase. It's going to move down to the genes, and then it's going to make the mRNA, and from there the ribosomes are going to start making the amino acids, polypeptide chains, it's going to start making those proteins, okay? And these are, in the trip operon, these are polypeptide subunits that make up enzymes that are going to be used for tryptophan synthesis. So going back to our, this diagram right here, this is that metabolic pathway to make tryptophan. And it requires 3 enzymes, okay? And those are the 3 enzymes that are coded for by these genes, okay? So there are 1, 2, 3, 4, 5 genes, and those 5 genes are going to help make those 3 enzymes. So what does the repressor do? What does the repressor do? This is the inactive form of the repressor. Okay, but remember how we said tryptophan is going to shut down those genes? Remember we said that? Well, it acts as a co-repressor. Remember that prefix co means together. It's going to work together with the repressor. Together as a repressor, when it comes together, it's now activated. It's now active. It's going to bind to the operator. Remember how we said that the operator is that switch? So this is the off. This is the off button, okay? When the repressor with the co-repressor tryptophan bind to the operator, it switches the genes off. And how does it switch the gene off? Well, the operator, this is the promoter right over here, and there's our RNA polymerase. The RNA polymerase, unfortunately, cannot bind to the promoter, or it gets blocked by the repressor with the tryptophan, and it can't get to those genes. Okay, so the genes are over here, and the RNA polymerase cannot access those genes because the repressor is essentially blocking it. It's a roadblock. The repressor is a roadblock, and it's blocking the RNA polymerase from getting to those genes. So this is how, it's a very simple mechanism that bacteria can utilize to shut off a gene. You have these repressor. The repressor is activated in the presence of a co-repressor. Together they bind to the operator, shutting down those genes, acting like a roadblock, preventing the RNA polymerase, this is the RNA polymerase, from accessing those genes, from transcribing those genes in transcription. Okay, so that was the trip operon. And the trip operon is an operon, so an operon that codes for, that includes genes that code for enzymes that are going to help the bacteria make tryptophan. Okay? And if there's enough tryptophan around, the tryptophan is going to act as a co-repressor. Okay? So that was the trip operon. Now let's talk about Lac Operon. You know, that lac stands for lactose, and lactose is a disaccharide, okay? It's a disaccharide. It's basically made up of 2 sugars, okay, glucose and galactose. And in order to break down or metabolize lactose, the bacteria need enzymes to break down that lactose, okay? So lactose, maybe I'll just draw it right over here. It's a disaccharide made up of glucose bound to galactose. So it's a disaccharide made up of those 2 sugars. And then those 2 sugars, in order to be digested, metabolized, and utilized by bacteria, they need to break that bond. They need to break that bond by using an enzyme. And also, remember that chemical reaction where we break bonds? Do you remember what it's called? Hydrolysis. Remember hydrolysis? We're breaking those bonds, okay? All right, so what's happening here is that in a Lac Operon, you also have the regulator gene, you have the promoter, you have the operator, and then you have the gene that's going to code for enzymes that are going to help break down lactose. Okay, and up here in this diagram at the very top, if there's no lactose around, the bacteria don't really need those enzymes, right? If there's no lactose around, why make the enzyme to break down lactose? So the operon is shut down. The gene is shut down, okay? And so what's happening when it's shut down is that the repressor is active all by itself. By itself alone, the repressor is active. It will bind to the operator. It's now off. The gene is now off because the RNA polymerase is prevented from being able to bind to the promoter and access the LAC-Z, LAC-Y, and LAC-A genes that are responsible for making enzymes that are going to help the bacteria break down lactose. Okay? So this is it off. The repressor is now on the operator. The gene is switched off. Why? RNA polymerase cannot get to those genes and transcribe those genes. Let's say that there is lactose around. Then the bacteria are like, oh, this is food. We can digest this lactose. So let's go ahead and start making these enzymes that are going to be used for breaking down lactose. So what's going to happen is that lactose, or in this diagram, it's called allolactose, which is a version of lactose. Okay, what's that word I'm looking for? Like I can't think of it right now, and I don't want to waste time. But allolactose is a type of lactose, a version of lactose. Now it's really going to annoy me that I don't remember. And it's called an inducer, okay, an inducer. And an inducer causes something to happen, whereas a repressor prevents something from happening. So with the trip operon, tryptophan was a co-repressor, where here in the Lac Operon, lactose is an inducer because it's going to induce, it's going to encourage, it's going to turn on the gene, okay? So we call the Lac Operon an inducible operon because it's normally shut off, okay? With the Lac Operon, it's normally shut off, whereas in the trip operon, it's normally on, okay? And then the tryptophan acts as a co-repressor, shutting it down. Whereas with the Lac Operon, it's usually off, and then with the presence of allolactose, which is a type of lactose, it's going to bind to the repressor, remove the repressor from the operator. Okay, so in this diagram right here, you can see it, the allolactose bound to the repressor. It causes the repressor to change shape, and it's going to pop off the operator, and now RNA polymerase has access to the promoter and can now access the genes. It's going to transcribe these genes. By transcription, mRNA is going to be made. By translation, now the amino acid chains are going to be made that are going to make these proteins, beta-galactosidase, permease, and trans-acetylase. Okay? So trip operon is a repressible operon, meaning that it's usually on, but then with tryptophan present, it's going to act as a co-repressor, turn off the gene. Whereas the Lac Operon is an inducible operon. It's normally turned off, but in the presence of lactose, it will be turned on. Okay? Definitely need to know these two operons for sure. So yeah, and this, again, this is just the picture. It's normally turned off, but when there is allolactose present, so let me just draw, like this is our allolactose, it's going to bind to the repressor, change the shape of the repressor will pop off the operator. Now then RNA polymerase is going to be able to move along the gene, okay, to the gene and transcribe those genes, and then those genes will be translated, etc. And then also, bacteria can digest and utilize lactose. However, the preferable source of food is actually glucose, okay? So bacteria can metabolize both glucose and lactose, but if lactose and glucose are both around, they will actually prefer for glucose to be metabolized. And so the gene that's going to help break down lactose will not be expressed as as much, because if it has glucose around, the bacteria will actually prefer it. Okay, so when glucose is scarce, meaning there's not a lot of glucose around, then the Lac Operon is on, and then the bacteria will be utilizing lactose. Okay, so here's an example of when lactose is present, but there's not a whole lot of lactose. When lactose is scarce, then CAMP, which is a type of nucleotide, CAMP levels are high. Okay, are high. So when glucose levels are low, CAMP levels are high. Okay, and so CAMP is going to bind to another protein called CRP, and together they're going to help RNA polymerase transcribe these genes. Okay, so when there's lactose present, it's going to bind to the repressor pops off the operator, and then in addition to that, CAMP is going to bind to the CRP protein, and it's going to help the RNA polymerase. I mean, I'm almost tempted to say that the CRP, the active CRP, is like a transcription factor, but it's not. Okay, prokaryotes do not have transcription factors, but you can think of it, it's almost like a transcription factor in that it helps the RNA polymerase bind to the promoter, and it promotes transcription. Okay, and in fact, it helps transcription happen a lot faster without the CRP, because without the active CRP, the RNA polymerase will transcribe the genes, but it'll be very slow and not a whole lot. Okay, so if lactose is present, but glucose is also present, then CAMP levels are going to be low. Okay, so if glucose is present, then the prokaryotes, they want to break down glucose. They prefer glucose over lactose. So these genes will still be turned on because lactose is still there. Okay, however, it's not going to be transcribing a lot of these genes because it prefers glucose over lactose. Okay. So in this scenario, lactose is present, just like the first scenario. However, glucose is present. There's a lot of glucose around. Therefore, CAMP levels are low. Therefore, the CRP is not active. It's not active. It will not bind to the promoter. It's not going to help the RNA polymerase bind to the promoter. And so transcription will take place, but it's going to take place very slowly. It says right here, RNA polymerase is less likely to bind, okay? So the active CRP activates, it helps the RNA polymerase bind to the promoter for transcription if there's only lactose around, okay? If glucose is scarce, then transcription of these genes that are going to help break down lactose is high, whereas if glucose is around, so is lactose, this operon is slowed down. Okay, it slows down because the CRP is not active. So when CAMP levels go up, it's going to help transcription. CAMP levels are only high. CAMP levels are only high when glucose levels are scarce. Okay, when there's not a lot of glucose, then CAMP levels are high. All right, so now we're going to talk about eukaryotic gene regulation, and there's at least 7 steps that are going to help regulate gene expression or gene regulation in eukaryotes. At least 7. It's much more complicated. With prokaryotes, it's a simple Operon model where there's a regulator that's going to bind to the operator, blocking those genes from being expressed. Okay, it's a lot more complicated with eukaryotes, and in the note paper that I put in Schoology, it has a table that has 7 different ways. It has 7 different ways that eukaryotic genes are controlled, on or off. Okay? So, these are the 7 ways. So these are the 7 points of control. Sorry, I'm a loud drinker too, so I was like, so I apologize if it's kind of an annoying sound, but okay, now I'm self-conscious about it. The control of gene expression can occur at any step in the pathway from gene to functional protein. So I'm going to actually move my face out of the way. Where can I move it? Maybe over here. Okay. I'm barely shown, but that's okay. You don't need to see me. So the first The first step is packing or unpacking DNA. You know, how tightly the DNA is coiled around the histone proteins is going to either shut down or turn on the gene, okay? Transcription, how fast transcription takes place, or if transcription takes place, mRNA processing, mRNA transport, translation, protein processing, and protein degradation. Those are the 7 steps for eukaryotic cells in which genes are controlled. Gene expression is controlled. Okay, so let's talk about packing and unpacking DNA first. So you know how we say that the DNA in eukaryotes is not naked, right? It's DNA that's wrapped around proteins, and specifically those proteins are called histones. And when they're wrapped around the histones, we call it a nucleosome. So this is a nucleosome. It looks like beads on a string. Okay, and then when you get chromatin, when you look at the DNA in the nucleus, it's in the form of chromatin. Okay, it's like loosely coiled DNA. Excuse me. And then when the chromosome, when chromosomes form, so chromatin's going to go into chromosomes in mitosis, right? I hope you guys remember what phase of mitosis you start to see chromosomes. I hope you guys are saying prophase, right? Prophase, okay? Prophase is when you start to see the chromosomes. But, I actually wanted, I want to talk a little bit about, you know, how does the DNA wrap around those histone proteins? You know, these histone proteins, there's like 8 of them, so you can count them. Here's a packet of 8. Those are the histones together. There's 8 of them that come together, 8 subunits that make up the nucleosome. 8 subunits come together, and they happen to be positively charged, and they're going to bind tightly to negatively charged oxygen. So the histone proteins are positively charged, whereas the DNA is negatively charged, so they're attracted to each other, and that helps with the packing of DNA. DNA is attracted to those positively charged histones. And I'm wondering if you guys remember why DNA would be negatively charged. When you look at the structure of DNA, it's made up of, you know, you have the deoxyribose sugar, right? So this is our sugar. You have the nitrogenous base. Remember, the nitrogenous base is like the adenines, the thymines, the guanines, the cytosine. And then you have the phosphate group. Well, here's the thing about the phosphate group. The phosphate group is surrounded by oxygens, and we've talked about this before, how these oxygens are electronegative. They have negative charges, okay? So they're sharing electrons with this middle phosphorus, this phosphorus atom, and it's unequal sharing, so they have negative charges, okay? So that is what gives DNA a negative charge, is the phosphate group, and it's attracted to the positively charged histone proteins, okay? So when DNA is tightly wrapped around histone proteins, and it's tightly wrapped around and coiled together in the nucleus. So this is a cell, and this is the nucleus of our cell. Can you guys see in the nucleus of the cell, there are dark spots, and then there's light spots. There's dark splotches and light splotches. Well, the darker splotches are called heterochromatin. And it's heterochromatin, it's tightly packed. That's why it looks so dense. It looks so dark because it's so dense. It's tightly packed. Whereas the euchromatin are the light areas, and it's loosely packed. You know, and if DNA is loosely coiled or loosely packed, then, you know, the RNA polymerase can get in there, can get into and can access the DNA and access the genes, and it can transcribe, and transcription can take place, okay? So heterochromatin, you know, those are where the genes are on, okay? So the genes are on because RNA polymerase can access those genes. They're not so tightly wrapped around the histone proteins, okay? They're not tightly packed together. Whereas the euchromatin is where the genes are actually, oops, I have it the other way around, Mishem. So if it's tightly packed, you guys erase what I just said, okay? Here we have the genes. Heterochromatin is where it's tightly packed, so the genes are off, okay? It's so tightly packed that the RNA polymerase cannot get in there. It can't get in there, and so those genes are not easily accessed by the RNA polymerase, so the genes are off. Whereas in euchromatin, it's loosely packed, it's loose or lighter DNA. Those are the genes that are on, okay? I'll have to listen back to what I just said, and maybe I can edit it. But anyways, heterochromatin, dark, tightly packed, condensed DNA. RNA polymerase doesn't have access to those genes. Genes are turned off. Euchromatin, lighter DNA, loosely packed. Those genes are turned on where RNA polymerase can access those genes. Okay, also related to DNA packing is that sometimes the DNA, the genes have methyl groups, methyl functional groups on them, okay? So these methyl functional groups, we call it methylation. When DNA is methylated, that means that the cytosine, remember the cytosine, which pairs with guanine, okay? The cytosine might have methyl groups. Oh, and it's wrong, it's not circled, but this is the methyl group. When cytosine, which is the nitrogenous base, is methylated, that's because there's a methyl functional group attached to it. Those genes are turned off, okay? So when DNA is methylated, those genes are permanently turned off. Once methylated, those genes are going to be permanently inactivated for the life of that individual or the life of the lineage of those cells, okay? When cells are dividing and you have the DNA genes that are methylated, those are going to be permanently turned off, okay? So no transcription. An example of this is in the X chromosome with the bar bodies. The X chromosome that becomes a bar body gets methylated. It gets methylated, and actually, in addition to being methylated, it gets tightly packed or coiled up. So RNA polymerase can't even access it anyway, okay? So that's one way that genes can be turned off, is if they are methylated. And I'm gonna have to pause the video here. I'm gonna continue after my meeting, okay? Okay, so this is where we left off. We were talking about how methylation is a way that genes can be turned off, okay? And this methylation is usually permanent, as I said. So when the cytosines on the gene is methylated, then usually it will be permanently shut down. And sometimes you can actually inherit these methylations. This is called epigenetic inheritance, where let's say your mom or your dad, they had a gene that was methylated. If you inherit the methylation, then it's a type of shutting down the genes that is not directly involving the nucleotide sequence, but it has to do with shutting down the gene, and that's called epigenetic inheritance, okay? And then also the histone proteins. The histone proteins can be acetylated, okay? The acetyl group, let me grab my pen. Okay, this is the acetyl group. It is COCH3, so carbon, oxygen, and then a methyl group. When those, when this functional group is attached to the histone, so you can see here, these are the histone tails with the acetylation, then it causes the chromatin to be loosely coiled. Okay, so acetylation of histone proteins causes the chromatin to be loosely coiled. And once it's loosely coiled, as we said before, then transcription can occur. The RNA polymerase can actually access the genes and transcribe the gene, and so gene expression will happen in areas where the histones are acetylated. Okay, and then also genes are going to be turned on when there are like transcription factors around. When the transcription factor assists the RNA polymerase to attaching to the promoter, then it's going to initiate transcription. So then here, the gene is going to be turned on, okay? And so we have the promoter, and then we have the transcription factors, and then we also have the enhancer sequence, okay? The enhancer sequence is it's upstream from the gene, so this is our enhancer sequence. If an activator binds to it, it causes the DNA strand to fold with the help of these folding proteins, and they're going to come near to the promoter, and together they're all going to work together to transcribe the gene. And it's going to enhance transcription. So this picture is showing you, here we have the enhancer sequence. You have the activators binding to the enhancer sequence, and then you have a DNA bending protein. These are some transcription factors that are all going to come together, working together to get that RNA polymerase to bind to the promoter and transcribe the gene. And in transcription, mRNA is going to be made. That's what we mean by RNA synthesis. And here's another picture of that happening. See, all of these proteins and all of these molecules are coming together to assist that RNA polymerase to transcribe the gene. And remember how we said RNA polymerase is the enzyme that's going to unzip and synthesize the mRNA, but it needs helpers. It needs helpers such as the transcription factors, you have activators, and in this picture you even have co-activators. So all of these molecules, proteins are coming together to assist RNA polymerase. And this is going to turn on a gene. This is the transcription complex that's going to turn on the gene because it's assisting the RNA polymerase. Okay? And so, with the activators that bind to the enhancers, what causes a specific gene to be expressed? It has to do with the specific activator in that cell. So we are comparing a liver cell with an eye lens cell, so the lens that makes up your eye, okay? They both have enhancer sequences. They both have the promoter sequence, okay? In the liver, you have the albumin gene that is going to be expressed. In the eyeball or the eye cell, you're going to have eye lens cells. You're going to have the crystalline gene that is going to be expressed. And you have these different activators. And these different activators in the different cells are going to activate, turn on whatever gene, okay, the albumin gene in the liver, the crystalline gene in the lens cell. So you can see here that the albumin gene is not expressed in the lens cell. The crystalline gene is not expressed in the liver cell. And so remember at the beginning of this chapter, the beginning of this video, I was saying that the question that we're trying to answer is how do cells become differentiated? It depends on which proteins are being expressed. In a liver cell, only liver proteins, associated proteins, are going to be made. And in the lens cell, only lens proteins are going to be expressed. And it has to do with what activators are available, okay? These activators are going to bind to the enhancers near that protein, causing near that gene to be transcribed, okay? So those enhancers near that gene, it's going to come together with activators and transcription factors and the RNA polymerase, and together they're going to work together to transcribe that gene that has to do with liver versus lens. Next, number 3 is post-transcriptional control. So, what protein is going to be made? Well, it depends on which exon is going to be considered an actual exon, which introns might be considered an exon, or what exons are going to be considered introns. Okay, so remember, we discussed this in the previous chapter. You can have like one segment or one gene, but you can end up with multiple proteins. And I think in your textbook, it even says that like over 90% of your genes actually can alternatively splice. You can get alternative splicing. So most of your genes are going to have alternative splicing. So this is a way of gene control, because whichever protein that your cells want to make, they can alternatively splice them. Okay, and since we have already went over alternative splicing, I'm not going to go over it again in this slide. Also, the amount of time that the mRNA is available to the ribosome is going to play a part in gene expression. Because if we have, let's say, an mRNA that can last for weeks, then a lot of proteins can be made from it in translation. So, after the pre-mRNA, the introns come out and the poly A tail and the 5' cap are added, and it goes out to the cytoplasm. This RNA, the mRNA that is mature, it can last anywhere between hours to weeks. So that has to do with gene expression control as well, because if it's only going to last for a few hours, the cell is controlling how much protein is actually being made. Okay. Ooh, that was very loud. Let me go ahead and lower the volume. Okay. So what determines how long these mRNAs last? There's two kinds of molecules. There's the si and the miRNA. And you guys they do the same thing. These are short segments of RNA that are complementary to the mRNA that is going to be destroyed. Okay. So the mRNA that's going to be destroyed, which has, an expiration date, let's say it's only going to last for 24 hours. The miRNA and the siRNA, these are short segments of RNA that are complementary to that mRNA. So here's our mRNA that's being targeted for degradation or destruction. Here's our siRNA or miRNA. They're going to bind to that mRNA strand, and they're going to cause proteins to cut up that mRNA into little pieces and destroy it. Okay, so here is a picture of an miRNA along with the protein complex, and here's our targeted mRNA that the miRNA is complementary to. And this protein is an enzyme. It's an enzyme that's going to degrade or cut up that mRNA. Or that miRNA with that protein complex, it's going to block, it's complementary to that mRNA, it's going to block the ribosome from translating it. Okay? So again, when we talk about gene expression, protein synthesis, this is an example of a gene being turned off after transcription. So transcription's taken place, then you have the mRNA, and to prevent the protein from being made, the gene being turned off indirectly by getting rid of, not even letting the protein being synthesized, you can get rid of the mRNA, or you can block the ribosome from translating the mRNA. Okay? So miRNA is a way for the gene to be turned off, not directly, but indirectly, by targeting the mRNA, by getting rid of the mRNA, or by blocking the ribosome, preventing the ribosome from translating that mRNA. Okay, yeah, so this is a brand new, topic. It's pretty brand new to our textbook, but this is just showing you how, that mRNA is degraded. There is a protein, called RISC, RISC, that dices up that mRNA in this example. In the miRNA with the protein is going to dice it up. But if it's siRNA, which is very similar to miRNA in that there's short sequences of RNA that target the mRNA, RISC is going to go ahead and degrade the mRNA. So yes, these two guys, they helped discover RNA interference. RNA interference. They were studying, I know that they were studying flowers, and then they noticed that some petals had white stripes on it, and then they discovered, the parts of the petal that had white stripes on it, their proteins for coloration were not being produced, and it was because of siRNA. They helped discover that. Okay, and then the fifth way that genes can be turned off is translation. This is similar to what we talked about before, where the mRNA is going to be blocked from the ribosome. Okay, so we can block initiation of translation if we have some kind of protein that's going to prevent the ribosome from binding to the mRNA. Okay, and then 6 and 7, protein processing. This is where genes are actually turned on. Okay, you turn on the gene by making it functional. Okay, by making this is supposed to be an arrow. Okay, by making the protein functional by adding. Let me just write functional. By adding sugar groups, or maybe adding functional groups, or maybe causing it to fold a certain way, cut a certain way, so that it can actually do what it's supposed to do. Okay, and then so this is number 6, and then number 7. This is for off, okay, turning the gene off. Let's say that the protein is now made, okay, so it's the gene has gone through transcription, the gene has gone through translation, the mRNA has been translated, and the protein is made, and the cell wants to turn off that gene by getting rid of the protein, okay, wants to get rid of the protein. There is what we call a death tag, okay, a death tag called ubiquitin, okay, which are these little protein molecules. These death tags will attach to that protein, and then it's a sign for the proteasome to come take that protein and cut it up and get rid of it, okay? It's sort of like ubiquitin is the death tag and proteasome is like the, the shredder, okay? It's going to shred up the protein. I remember a student long ago was like, so ubiquitin is like, they hire... they hire someone to take out another person, and the proteasome is that person that takes them out. I guess you can think of it that way, okay? So ubiquitin is the death tag, and then the proteasome is the enzyme. It's the enzyme that's actually going to break down that protein, okay? So it's interesting because, like, a protein is made, so, the protein is made, the gene went through transcription, and then the mRNA was translated. A ribosome made that polypeptide chain, and then it went through protein processing, either folding or cleaving or whatever. But then the last step to turn off the gene would be protein degradation, where ubiquitin tags it to be chopped up, to be degraded into like short amino acid sequences. Okay, so yeah, ubiquitin was discovered in the 1980s, so it's more of a recent thing, and this is what proteasomes look like, okay? So as I was saying, that there are several instances in the eukaryotic gene control system where genes can be turned on or off, and it can be direct on the gene or it can be indirect. So after transcription, after translation. So just as a summary, right? Chromatin modification, you can have acetylation, you can have methylation. So methylation turns off, acetylation turns on, right? Transcription, you have to have all the activators coming to the enhancers and then transcription factors coming together with the RNA polymerase to turn on the gene, okay? And then you might have different alternative RNA splicing that's going to turn on one gene versus another, and then it's translated, or translation is blocked, that's turning off, and then or maybe the mRNA is degraded before translation can occur, and then the protein, if it is made, it can be degraded, etc. Okay? Okay, so yeah, this is the whole story. There's 7 in this presentation. There's 7 ways that genes can be turned on or off in eukaryotic cells, okay? But remember, in prokaryotic cells like bacteria, it's just 2 or 3, okay? Okay, and then the last section of our chapter in the textbook is like how, like, besides turning on or off genes, how, how are organisms programmed for differential gene expression? So here we have like the metamorphosis of a frog, right? It's going from a fertilized egg, which is a single cell, to an embryo, to a tadpole, to a froglet, okay? And then it becomes a frog. So how does that all happen? Well, so the genetic program for embryonic development is transformation from a zygote to an adult, and it happens in 3 ways: by cell division, cell differentiation, and morphogenesis, okay? So when we go from a zygote, a single cell, it's dividing by cell division, right? You go from 1 cell to 2 cells, and then from there, the ball of cells becomes, you go from 2 to 4, and then it just becomes eventually this ball of hundreds of cells. Oops, I just moved my screen over there, okay? To a ball of cells. And once it becomes a ball of cells, we call that a blastula, okay? A blastula. And it doesn't have any shape yet. It will take shape, and that's going to happen through differentiation and morphogenesis. Morphogenesis means to take shape. Morph means to form. Okay, and genesis is like taking shape, the beginning of taking shape of form. Okay, and we're going to be talking about cell differentiation as well. Let me go ahead and move my little picture out of the way. So what generates the first differences among cells in an early embryo? Okay, so here we have our egg that is now fertilized. It's a single cell zygote. There's something called cytoplasmic determinants. There are proteins and mRNA. So mRNA or proteins, those are cytoplasmic determinants in the egg. And can you guys see in this diagram that it's unequally distributed through the cell? Like I see more of these, round proteins versus this triangular proteins. I see more of the round on the left side. I see more of the green triangle proteins on the right side. So these are called cytoplasmic determinants, and it's unequally distributed in the cell. And that's actually important because where these cytoplasmic determinants are found in the first cell is going to determine where is the head going to go for this organism. Maybe this is a frog. Where is the tail of the frog going to end up? Where is the head? The body axis is usually determined by these cytoplasmic determinants, okay? And I'll give you guys an example of that. We have mRNA of a protein called bicoid, okay? So bicoid is a protein, but the mRNA of bicoid protein is a cytoplasmic determinant, okay? And so here is a egg, and you can see this is a microscopic photo, and then this is the drawing of that egg, okay? And at this point, it's not fertilized, but you can see this dark area on the far left-hand side. That's where there's a lot of bicoid mRNA. So that mRNA is ready to be translated, right? It hasn't been translated yet, but it's gone through transcription. And so when you see a lot of that mRNA on that side of the egg, once it becomes, fertilized, then translation is going to take place of that mRNA. And so that bicoid protein is going to start being made on that left side of the zygote. And once that happens, this is going to determine the anterior end of that organism. Okay, so the anterior end of like the fruit fly. So what we're talking about is the fruit fly here. So anterior means the head, and then posterior means the end, basically where the butt is. Okay, so the head is on this side, posterior is on this side. So this is a cytoplasmic determinant. The bicoid mRNA, wherever you find a high gradient or high concentration of bicoid mRNA, which is going to lead to mRNA, which is going to lead to the bicoid protein by translation. If there's a lot of bicoid protein on that side of the egg, then that's where the head is going to form. Okay, so what's happening here is that you start off with no form. You start off with just a plain egg, but then eventually it's going to take form, and that's called morphogenesis, where the embryo is taking form. It's going to eventually grow a head, a butt, arms, legs, wings, etc. Okay, so the bicoid protein is called a morphogen. I love these words. Morphogen, because it's going to be a molecule that's going to give that organism form, because morph means to form. Okay, so it's called a morphogen because where it is found will determine the anterior end or the front end of the fruit fly. So the second way, so this was the first way that, this is the first way that, you start to see differences in embryos. You have the cytoplasmic determinants. And then the second way is by induction, where signal molecules from nearby cells causes changes to the target cells. So let's say that this is our target cell right here. So this is our target cell. And then this is the neighbor cell. Okay, the neighbor. So they're just, nearby, okay? The neighbor cell is going to release these signal molecules. They're going to bind to receptor proteins. And by signal transduction pathway, do you guys remember we talked about that? Okay. By signal transduction pathway, it's going to cause a reaction within the cell. Let's say it's going to turn on, okay, it's going to turn on genes that are going to cause this target cell to become a specific type of cell. Let's say that this neighboring cell, let's just say that this neighboring cell is an epithelial cell or like a skin cell. Epithelial. And so it's going to release these signal proteins that are going to cause the target cell to become also an epithelial cell. Okay? So I always tell my students, like, induction, it's sort of like peer pressure. It's pressuring the neighboring cells. It's pressuring a cell to become a specific type of cell, right? So like if you hang out with like wrong crowd, you're gonna, do the wrong things, right? So induction is where signal molecules from embryonic cells causes changes in nearby target cells. So induction. And a good example of this was done in an experiment a long time ago. These two scientists, what they did was they took in I believe this is a newt, okay, a newt or some kind of amphibian, okay, a newt or amphibian. This is a blastopore. It's a ball of cells, okay, and there is, as it's developing, as it's taking form, there is this one area of our blastula called the dorsal lip. The dorsal lip of the blastopore. By the way, this is all a part of embryology. And I took an embryology course back when I was in college, and to be honest, it was very complicated. There was so much going on in the development of animals. Like, we had to memorize the development of fruit flies, frogs, humans, chickens. Those are like the four main organisms that we had to study and memorize. But this is of a newt, like frog or some amphibian, okay? So the dorsal lip of the blastopore, what that part of the blastula is going to end up being the dorsal side of that amphibian. Dorsal means the backside, like a dorsal fin. Think of like a dorsal fin of a of a dolphin. That's where the spine is, okay? So the dorsal lip is going to become the spine. That area is going to become the spine of our newt or frog. So what they did was they removed the dorsal lip from one blastopore and they transplanted it to another newt or frog on the opposite side of its blastopore. Okay, the dorsal lip of the blastopore. So now this blastula, which is forming into a gastrula, this gastrula has two dorsal lips. It has two dorsal lips. What does that mean? Remember how I said the dorsal lip of the blastopore turns into the backbone, or the backside, the dorsal side? Well, guess what? What happened was they allowed for this newt or frog to develop, and it ended up looking like this, where now you have two dorsal sides, okay? You have a dorsal side there, and then you have a dorsal side there because it had two blastopores, dorsal lips of the blastopore. So what I'm trying to say here is that by induction, by neighboring cells, it's going to cause those portions of the developing embryo or gastrula to turn into a specific body part. So it released proteins to neighboring cells to cause it to become the dorsal side. Okay? So this is an example of induction where you have cells releasing proteins, influencing the neighboring cells to become a certain type of cell, and in this case to become the dorsal side of the embryo. So because there were two dorsal lips, it ended up with two backbones. Okay. So cells, when they turn into different kinds of cells, like muscle cells, fat cells, bone cells, blood cells, nervous cells, that happens, that's called differentiation. But before differentiation, there is a step before differentiation. Before a cell becomes a different type of cell, it has to be determined. I mean, think of it this way, when you become a doctor, when you become a teacher, you get a job. That's called like, that's like differentiation, right? We all get like different jobs, different labels. But before you can do that, you have to make that decision. You have to be determined to become a doctor, determined to become a fat cell, determined to become a bone cell, a blood cell, etc. Okay? So determination is irreversible. It's when a cell commits to become a particular type of cell. And how does that mean on a cellular level? That just means that certain genes are going to be turned on. So determination equates with which genes are turned on. So if in a muscle cell, let's say that the stem cell, okay? So we have a stem cell. When those genes that are associated with muscle protein, when those genes are turned on, then it's determined to become a muscle cell. And when they start making those proteins, that's when it becomes differentiated. Okay, differentiation. So differentiation is when the cell starts making those tissue-specific proteins, but determination is when those genes are going to be turned on. So you turn the genes on, and then you make the protein. So turning the gene on is determination, and then differentiation is when the proteins are actually being manufactured in that cell. Okay? A good example of this would be muscle cells. Okay, muscle cells. And I kind of talked about it just on the previous slide, but how does it happen? Okay, so here we have, just a stem cell. Okay, so this is our stem cell here, and every cell has these genes. Okay, because in all of our cells we have muscle-specific genes, but the only cells in our body that have these genes turned on would be muscle cells, right? There is a regulatory gene called the MyoD. Okay, it's called the master regulator, because once it's turned on, then it's going to cause the cell to become a muscle. So let's say that this is our stem cell, and then it becomes determined, meaning that those genes have to be turned on. Okay, and it turns on when the master regulator gene is expressed. And the master regulator gene, it actually codes for a transcription factor protein. It codes for transcription factor. And that transcription factor is going to turn on the other 3 genes that are associated with muscles. Okay? And so then that cell starts to make muscle proteins. And that's differentiation. Okay? So determination, this is an example of determination of when the genes are turned on. You have these transcription factors, or the transcription factor, the master regulatory gene is turned on, which is going to assist in the transcription of these other proteins. Once these proteins are being made, then it's a differentiated muscle cell. So when we look at the fruit fly, when we look at a frog, when we look at us, okay, we are what we call bilateral organisms, meaning we have a left side and a right side, and we have a body plan. Okay, our body plan is bilateral. We have a left side and a right side. Like you can split down the middle, left, right side. And the body plan, how it was set up, how it was formed, is called pattern formation. Pattern formation is a development of spatial organization of tissues and organs. Like, where is everything going to go? Where is the head going to go? Where are the eyes going to go? Where is the hands going to go? Where are the feet going to go? Where's the heart, the intestines, the stomach? Where is it all going to go? That's called pattern formation. And what's very important about pattern formation is positional information. And we're going to be talking more about this when we go over like cell signaling at the end of the semester. Positional information an example of positional information is the cytoplasmic determinants, because those proteins or those mRNAs give cells an idea of like what they're going to become. Like, remember we said that the bicoid mRNA, if it's found on one side of the egg, that's positional information that's going to tell that the zygote that's the head. That's the side where the head is going to be. The anterior side of the organism. So here we have anterior, posterior. So this is the anterior part of the fruit fly. This is the posterior part of the fruit fly. And then up here we have the dorsal side. We have the ventral side. And then you have the left side, the right side. So in a bilateral organism, you have anterior, posterior, dorsal, ventral, left, right. Okay? And where it all forms is that formation is called pattern formation, and it starts with positional information such as cytoplasmic determinant. It's going to determine where is the anterior, where is the posterior, where is the dorsal, where's the ventral. Okay. So yeah, here is just a diagram showing you, like in a fruit fly, you start off with an egg, right, and then you have the bicoid mRNA is going to be on this side because that's going to be the head. That's supposed to say mRNA. Okay. That's the bicoid mRNA, which is a cytoplasmic determinant. And then it just starts to go through body segmentation. And then here we have a larva. And then eventually it's going to turn into a mature fruit fly. And there are genes that are responsible for this. There are genes that are responsible for the head forming, the abdomen, the legs, the wings, etc. And these genes are called homeotic genes. Homeotic genes. I want to show you this picture of a fruit fly head because this is a normal wild type head of the fruit fly. Whereas over here, this is the mutated fruit fly head. Here you have just normal antenna here, okay? But over here, unfortunately for this fly, in the laboratory, what scientists did was they turned on the wrong genes in the wrong place. They turned on the wrong genes at the wrong place, and instead, this fruit fly was born with legs instead of antenna. Okay? So, pattern formation, right? It's like the spatial development of organs and tissues in the right place. And with these homeotic genes, you can turn on the genes in the wrong place, and you're going to get the wrong body parts going in the wrong place. But if they're turned on in the right place, then you're going to get the correct body pattern formation. So homeotic genes, these are groups of genes that control pattern formation, body axes, organization of where tissues, organs, or all the parts go. Okay. So for example, in a fruit fly, on a chromosome, they're going to have 1, 2, 3, 4, 5, 6, 7. These are just 7 homeotic genes. And let's say that this gene right here, it's the lab. I think that's a LAB. Okay, LAB gene that is going to be responsible for making the head. And you can see that it's turned on in that body segment of the embryo. It's turned on in that body segment of our embryo, which is going to eventually lead to the head. Okay, and then also the DFD gene has to be activated. This gene needs to be activated in that segment of the embryo in order to get the mouth of the fruit fly's head. Okay, so all of this is a color coordinated, diagram that shows you how these homeotic genes, they need to be turned on in the right place, or else you're going to get the wrong body part growing in the wrong place. Okay, it determines the segments of the adult fly. I believe like all mammals, most animals have Hox genes. So Hox genes are a type of homeotic gene. Okay, I just talked about homeotic genes here, but Hox genes are an example of homeotic genes. So here was the fruit fly Hox genes. Mouse and humans, you guys, we share a lot of Hox genes with mice. That's why we can have like mice grow. We can do experiments on mice, and we can kind of come to, we can use them as models for understanding how human embryos develop as well. Okay, so in humans and mice, we have a lot of similar Hox genes. Okay, we'll be talking about paralogous genes. There's analogous genes and paralogous genes. We'll be talking about that when we go over evolution. Okay, because these Hox genes, because we have a lot in common with even fruit flies, that would be an indicator or points to an ancestral gene. Okay. Oh, I found this picture online. This is an example of an experiment that was done on fruit flies where they turned on the wrong gene in the wrong place. So the PAX6-HOX gene, this is a HOX gene that controls eye growth in the fruit fly, mice, and humans. Okay, in humans. And when you turn on that gene in the wrong place, you're going to get eyes developing in the wrong place. So they did this experiment where they turned on the PAX gene in mice in the knees, and they found eye tissue growing in the knees. Here's a picture of Drosophila, or the fruit fly, where the knees, you know, of the legs have developed eye tissue. Okay? I mean, it doesn't mean that the fly can see from, you know, use all of these eyes. It can't. Okay? They're usually non-functional. However, the eye tissue is growing in the wrong place. So that was an example. Okay, so what genes are responsible for pattern formation? Homeotic genes, and also maternal effect genes. And we've already talked about the bicoid gene, which is going to give body axes information. Where is the anterior or posterior end of our organism? Okay, so the maternal effect genes, they encode for the cytoplasmic determinants, like the, you know, the bicoid protein. Okay, so the gene for bicoid is a maternal effect gene, which is going to initially establish the axes of the body of the Drosophila, or the fruit fly. Anterior versus posterior. Okay, so where you can find the bicoid mRNA, where you find it in high concentration, that's going to determine the anterior end of the egg, okay, or the zygote. And here's a picture of an experiment that was done. And you know, that's how a lot of scientists figure out what things do. This is how we can figure out what genes do, is if you can knock them out. If you can knock out a gene, if you can mutate the gene, and then you let the organism grow, and you look at the development, or like, you know, what is disabled. Sorry, you guys, there's like noise going on in the back. Whatever is disabled about the organism tells us what that gene does. Okay, so what they did was they took the, you know, the bicoid mRNA, and they put some on the other end. Okay, they put some on the other end. What happened was that, so this is normal at the top. This is the wild type, where you have a lot of bicoid. The gene is activated in the anterior portion, okay? And so when the gene is activated, the mRNA is made, and that becomes the head. Well, what they did was they caused the proteins to start being expressed. I'm sorry. They knocked out the gene. So it wasn't being expressed on this end. So you end up with two tails. Okay, so that's why I was talking about knockout. It's because they knocked out the gene. So you end up with a mutant larva with the knockout bicoid, meaning they don't make the bicoid mRNA. So no bicoid mRNA on either side of the egg. You're going to end up with two tails, no head, no anterior portion of our fruit fly. Okay? I think that might be it. Yeah, so chapter 18. That was really interesting. And I hope that the video was interesting for you guys, and I hope that you guys learned a lot. Okay?